So the recording of the discussion is actually hidden or unlisted kasi hindi sa akin yung slides. These were the slides I use in another university that I taught. Minodify ko lang siya. So for copyright concern. But the contents of the PowerPoint naman is found within the activity sheets nyo. So you could use that as a reviewer. So next week class, that would be on February 3. Mag-meet tayo on site. We'll do activity 2 and we'll have your quiz number 2. Yung quiz number 3 nyo sa precipitation, isasabay ko na lang siya sa exam nyo. After the discussion today, I'm gonna explain to you what you're going to do for activity 3. Yung activity 3 kasi class hindi na natin siya magagawa on-site kasi wala yung Petri dish na kailangan dyan. Walang stock yung lab tech natin, yung instrumentation room natin. Very small Petri dish kasi yan kaya hindi natin siya magagawa. Last academic year nung ginawa namin yan, umorder pa ako sa Lazada. Hassle pa kung pabibilin ko pa kayo. Anyway, let's have the discussion of your precipitation. Now in the activity 2, we talk about agglutination, di ba? This time naman, precipitation tayo. If in agglutination, it would usually involve antigen and antibody. But for precipitation, it involves antigen and a soluble substance and a antibody. So sa agglutination, dalawa lang, usually antigen, antibody. But for precipitation, tatlo sila. It would involve another substance, yung mga soluble substances, which you will learn in this activity. So with that, let's start your activity 3, precipitation discussion. Meeting class, I'm going to discuss precipitation. Now, precipitation class would require the formation of a visible lattice that would result from the combination of soluble antigens to its soluble antibody. Diba, if you would compare precipitation versus agglutination, so agglutination you would be using cellular antigens. So it's very easy to form agglutination. But when it comes to precipitation kasi class, you would be using soluble antigens. So it's very difficult to form a type of attachment to an antigen to antibody. So you would be using different substances or components to form this for lattice formation and attachment. It would usually work best class when antigens and antibodies are at an optimal proportion, wherein there would be a zone of equivalence. So ang precipitation class, they would work best if you have equal amounts of antigen and antibody. If you apply this in the lab, dapat yung volume ng antigen mo, yung volume ng antibody mo equal amount. Katulad nung nangyari sa atin sa lab class. We had a difficult time of creating an agglutination because we were not able to do a zone of equivalence. Mas madami yung volume ng RBC antigen nyo kaysa dun sa serum which would contain antibodies. Nag-cause ng false negative due to post-zone and pro-zone reactions. Now, precipitation class would occur in 2 steps. Number 1 would be the rapid invisible formation of antigen antibody aggregates. While the second one is that the slow forming visible complex of lattice of interlocking aggregates class. So take note of that. So the first one again is the rapid invisible formation of antigen antibody aggregates. So sa precipitation, ang first step is that your antigen would attach to your antibodies or your antibodies would attach to the antibody. Then the next one would be the lattice formation. Mag-interlock class. Mag-a-attach yung antigen-antibody, yung aggregates, yung antigen-antibody aggregates with one another, forming this so-called interlocking aggregates. So nag-combine siya class yung mga antigens and antibodies nyo together. Now, just a recap class, there's a so-called precipitation curve. So this is similar with your agglutination. Yung pro-zone zone of equivalent, post zone. Now take note of this class. A pro zone, again, would be wherein the reaction would have an antibodies that are in excess. There is insufficient reactive sites on antigen for lattice formation. So sobrang dami ng antibodies class. So much antibodies, this would cause insufficient reactive sites doon sa antigen. Therefore, magkakos siya ng false negative. While if there is a post zone naman class, this time antigens are in excess. Meaning class, there would be insufficient reactive sites naman on your antibodies for lattice formation. So walang kulang yung reactive site sa antibodies nyo due to having too many antigens. And the best one, the zone of equivalence class. This is wherein the concentration and ratio of both antigen and antibody are equal or at equilibrium. Here class, there would be the formation of a precipitate. Both prozone and postzone class would again lead to false negative results. Number one class is precipitation in solution. So there is what you call a ring precipitation test, flocculation tests. Under flocculation test, we have your rapid plasma reagent tests. Now, the instrumental methods to enhance sensitivity in measuring precipitation class are the following. So you could perform turbidimetry and nephelometry. Then we have your precipitation by passive immunodiffusion. Under passive immunodiffusion, we have your linear immunodiffusion. This is a single linear immunodiffusion known as your Udin technique. Then we have your double linear immunodiffusion technique known as your Oakley-Fulthorpe immunodiffusion. Then we have your radial immunodiffusion. Under radial immunodiffusion, we have your single radial immunodiffusion, also known as the Mancini and Fahey technique. Then we have the double radial immunodiffusion, also known as your Auchter-Loney technique, which later class i-discuss natin one by one. Then there's also the precipitation by electrophoretic techniques. We have here your electroimmunodiffusion. Under electroimmunodiffusion, we have your one-dimension electroimmunodiffusion, such as your single electroimmunodiffusion, known as your rocket immunoelectrophoresis. We also have your double electroimmunodiffusion, known as your counter immunoelectrophoresis. Then we have your two-dimension electroimmunodiffusion, known as your cross immunoelectrophoresis. Then we also have immunoelectrophoresis and immunofixation electrophoresis. Let's start with the precipitation in solution techniques class. Number one is the ring precipitation test. Now, the ring precipitation test class is usually performed in a tube and would require the formation of an antigen-antibody precipitate between two fluids as a ring of precipitate that is layered on the surface of the antibody. If naalala niyo yung COVAX test nyo sa bacteriology, ganyan yung itsura niya, class, di ba sa COVAX, pag nagpataka ng reagent, magkakaroon ng parang ring dun sa top. Now, dito sa ring precipitation test is that there would be the formation of precipitate, an antigen-antibody precipitate. Ito yung tatandaan nyo yan. There would be antigen-antibody precipitate in between two fluids. So what you would first do, class, is that you would add your serum, and in the serum class, there would be the presence of antibodies. After adding the serum, so ito yung serum nyo. Kunwari ito yung serum nyo. This would contain the antibodies. After adding the serum that would contain the antibodies, you now add your antigen. Now, what happens is that your antigen would be attracted to the antibodies. Similar class yung antibodies nyo would also be attracted by the antigens. Therefore, since both of them are attracted together, they would meet halfway sa gitna forming a zone of equivalence or a visible precipitate at the middle. Kaya siya tinawag na ring precipitation test. So do take note of that, class. There's a zone of equivalence or a visible precipitate at the middle. Then we have your flocculation test. Now, flocculation test, class, may be performed in a slide or a tube. In this test, a drop of antigen solution is added to a drop of patient serum on a slide, and the result is recorded after shaking the slide. In a positive test, class, the floccules would appear, which can be seen by naked eye or demonstrated well under the microscope. Example of this, class, would include your RPR test for syphilis, which would use a RPR card. So I believe meron tayong stock ng RPR card, class, sa lab. Maybe we could perform this in the finals. Then we have your VDRL test for syphilis, which would use a slide, and the CAN test for syphilis, which would use a tube. So let me repeat, class, yung procedure nito, you would add a drop of antigen solution to a drop of patient serum on a slide. Then you would wait for precipitation to occur by shaking the slide. Floccules would appear, class. Parang buo-buong parang buo-buong clots. So ito siya. So here you would have the serum with the reagent antibody. You would centrifuge it, then precipitate would form, then after shaking, magdi-disintegrate yung precipitate, forming these flocculates or floccules. Then for a negative flocculation, class, here we have your serum with reagent body. You centrifuge it, it would form a precipitate at the bottom. Then you shake it, there's no flocculates. Walang flocculation na mabubuo. It's still a whole intact precipitate. Then we have your rapid plasma reagent test, so your RPR. Now, RPR, class, is a non-treponemal serologic test for syphilis. When you say non-treponemal, class, you do not use your treponema pallidum antigen. Instead, class, what you use here is a antibody known as your reagin. Reagin yan, sorry, class. REAGIN. Your reagin is an antibody formed against cardiolipin during the progress of the disease. The reagent antigen would consist of cardiolipin, lecithin, cholesterol, and 10% chlorine chloride, EDTA, and charcoal. So here's the procedure for your RPR test. So cardiolipin would coat the charcoal particle. Di ba nga pag precipitation, class, you would use certain particles. In this case, ang ginagamit yung particle is yung charcoal. So cardiolipin, which is the antigen, would coat the charcoal particle. You now add your patient serum that would contain IgM and IgG against your cardiolipin reagent. Now, let's say class positive, merong syphilis yung patient niya. Positive siya for syphilis. So since positive siya sa syphilis, the patient would develop antibodies against certain components of the syphilis, specifically cardiolipin. So what happens is that the antibodies of the patient positive for syphilis would attach to the cardiolipin coated with charcoal. And that would form your precipitation. So take note of that, class. Yung antigen na ginagamit nyo dito sa RPR is yung cardiolipin coated with a charcoal particle. So ito yung RPR card na gagamitin natin sa lab. So what you would do for every card, class, you could do up to 10 tests. So first thing you need to do, you would add your serum. After adding your serum, class, you add your cardiolipin reagent. After adding the reagent, you would rotate it for 8 minutes on a rotator at 100 RPM. So ulitin ko, you add your serum, Usually 1 drop of serum yan, class, plus 1 drop of your reagent, your cardiolipin reagent. After that, you would rotate it for 8 minutes on a mechanical rotator at 100 RPM. Read macroscopically while in the wet state under a high intensity incandescent lamp or strong daylight. When I say wet state, class, hindi pa nag-dry yung combination ng serum sa reagent nyo. If it becomes dry, class, magkakaroon ka ng hindi na siya viable for reading. So you have to make sure, class, that it is in a wet state. So this is the positive reaction, class. Take note of the formation of precipitates. Then this is a negative or non-reactive reaction. Now let's talk about the instrumental methods that would enhance sensitivity in measuring precipitation in solution, yung nephelometry sa turbidimetry. Let's start with your turbidimetry. So this is more of a review, class, sa instrumentation nyo sa CC1. Now the turbidimetry, class, what is being measured is the initial cloudness. It is measured, class, by passing a light through the solution and determining the amount of light that would come directly across the solution into the detector. Halimbawa, ito yung tube nyo. Let's say, ang gagawin is that sobrang turbid niya, class. You would now expose this to light. Now, yung light, class, would pass through the solution, and you would measure. I-measure mo yung amount of light able to pass through. So kung mas turbid yan, mas turbid yung solution nyo, the less light that would be able to pass through. While if it's clear, the more light that would be able to pass through. So that's your turbidimetry. This is a measure of amount of light that is lost due to scatter by the lattice structure. So yung amount of light, class, that is not able to pass through the solution, it means that the light is being scattered by the lattice structure. The amount of scatter, class, is proportional to the concentration of molecules in the lattice structure. So here you would have your laser light. It would pass through the solution. Then it would be measured by a detector. And take note of this, class. Ulitin ko, the amount of scatter is proportional to the concentration of the molecules in the lattice structure. Then we have your nephelometry. Now, this is somewhat similar, class, with turbidimetry. The difference, class, is that it does not measure the light directly across. If you take a look at turbidimetry, class, yung pag-measure ng light niya is across the solution. But in nephelometry, class, you would measure the light in a different position, specifically at a 10 to 90 degree angle, typically 70 degree angle from the light source. So instead of having a straight line similar with your turbidimetry, you would have the light source positioned at an angle of 10 to 90 degrees, typically 70 degree angle. Then you would measure the light. Now let's go to the precipitation by passive immunodiffusion. In passive immunodiffusion, class, this method would allow the combination of specific antigen and antibodies in a semi-solid gel or media. An example of a semi-solid gel or media is your agar. So you would identify the combination of antigen and antibody using gels or media such as your agar. Now, performed using agar or agarose gel, it would stabilize the diffusion process and would allow visualization of precipitin bands. So magkakaroon ka ng precipitation bands, which later I will show to you. It does not use an electric current to diffuse. Diba sa electrophoresis nyo? Sa electrophoresis nyo, you would use an electric current to diffuse different types of proteins. If naturo sa inyo sa Molbayo nyo class, yung purpose ng electrophoresis nyo is to separate proteins based on their molecular weight. The smaller the molecular weight of a protein, the faster it would be, the farther it can be. So yun yung purpose ng electrophoresis nyo. So hindi ka, in this method, you do not use an electrophoresis to diffuse your antigens and your antibodies. Now, this reaction in gel immunodiffusion method is stable class compared to precipitation in solution. The reason why it's more stable class is because you're using a semi-solid media. Compared with having it done in a solution katulad ng ring layer kanina, mas stable to because you have a fixed media or you use a fixed support. Now, immunodiffusion reactions are classified according to the following. Number of reactants diffusing, known as a single or double, and direction of diffusion. Direction of diffusion class, it could be one-dimensional or two-dimensional. Pag sinabi mong direction of diffusion class, they would have a movement of diffusion. Kung paano sila kumalat, there's a movement of their separation or diffusion. Pag sinabi mong one-dimensional class, this would include the following techniques. Single linear immunodiffusion, also known as the Udin diffusion. Double linear immunodiffusion, which would include Oakley and Fulhorp diffusion. Then we have your two-dimensional single radial immunodiffusion, known as the Mancini diffusion and Fahey diffusion. Then we have your double radial immunodiffusion, known as the Austerloni double diffusion. Now let's talk about the direction of diffusion. Now pag sinabi niyong one-dimensional, the movement of the antigen or antibody would follow a vertical or horizontal direction of diffusion. That's why the precipitin line is linear. Pag sinabi mong linear class, it could only be straight or vertical, or it could be horizontal. So either. So yun yung reason kaya siya tinawag na one-dimensional. It can only come in a single direction. It could be vertical or horizontal. Kapag naman two-dimensional diffusion, it could come as either vertical and horizontal. Let's say, ang possible result ng movement ng lines mo would become like this. Or pwede ding ganyan. So that's the two possible result in a two-dimensional diffusion. That's why the precipitin line class would come as radial or somewhat diagonal. So take note of that, ha? So ulitin ko, ha, pag one-dimensional, it would only come as a single direction, either vertical or horizontal. But if it's two-dimensional, it would come into two directions, both vertical and horizontal, forming the precipitin line to become radial or parang diagonal. Now let's start with the single linear immunodiffusion. Now single linear immunodiffusion is known as your Udin diffusion technique. The antibody class is incorporated in an agar gel in a test tube, and then the antigen is layered over it. So ito siya. So here you would have your tube. Sa tube nyo class, you would add your media or your gel. In the gel class, it would already contain the antibody. After doing that class, you now add your antigen. Again, diba, they would be attracted to one another. Antigen would be attracted to the antibody. Antibody would be attracted to the antigen, forming a precipitin band at the middle. Ito siya. This is your precipitin band. At the middle of the gel, of your antibody gel and your antigen. Pag nagkaroon ka ng precipitin band, it means it's a positive result. If you take a notice, if you remember class, 'di ba kapag single immunodiffusion siya or linear immunodiffusion siya, there's only one direction. In this case, ang precipitin band nyo came in the form of a horizontal direction. So it's similar with your ring layer test, but this one is more stable since you're using a gel or a media. The antigen would diffuse downward through the gel, forming a line of precipitation. Precipitation dissolves as the concentration of the antigen increases. The number of bands indicates the number of different antigens. Then let's go to the double linear immunodiffusion. This is also known as your Oakley-Fulthorpe diffusion technique. Now, antibody is incorporated in the gel above which is a column of plain agar. So kanina class, sa Udin technique nyo, yung antigen nyo would come in as a solution. This time, instead of a solution in the Oakley-Fulthorpe, you're going to use both media or agar. The difference lang is yung first agar layer nyo. The first agar layer would contain the antibody, while the second layer or the second agar would be plain. So antibody is incorporated in the gel above which is a column of plain agar. The antigen is layered on top of the agar media. So ito yung antibody which would contain, which will be found in the agar gel. At the middle of this class is the plain agar. After that, you add your antigen solution. So mag-attract yung antibody and antigen, forming a precipitin band in the plain agar. In this case, class, both antigen and antibody were able to diffuse towards the middle in a horizontal, forming a horizontal precipitin band. The antigens and antibodies would move toward each other through the intervening column of plain agar. This will result in the formation of a precipitate when they meet at optimum proportion. Now let's go to the single radial immunodiffusion. Now in this type of immunodiffusion class, the antibodies are spread homogeneously in an agar media, and the antigens would diffuse within the agar. Here, the gel agar would contain diluted antibodies specific to certain antigens. A ring of precipitate would form upon the addition of the antigen into the well, which is cut in the agar. So kunwari class, ito yung agar nyo. Ang gagawin nyo dito this time is that you would cut into the agar. Magka-cut ka ng isang ring shape dito sa gitna. And that is where you would add your antigens. Magpo-form ka ng parang well. Inside that well, you would add your antigens. So ulitin ko class, you're going to use here agar that would contain antibodies. After adding the agar, letting it solidify, semi-solid na siya, you cut a hole. Cut ka ng hole or gagawa ka ng well sa agar. And that is where you would add your antigens. The ring would form at the equivalent point between the antibodies and the antigen. The diameter of the ring would depend on the antigen concentration. Thus, the higher the antigen concentration, the greater the diameter of the ring. So pag gagamit kayo class ng very concentrated antigen amounts or antigen sample, you need to make sure that your ring would also be, or the diameter of the well or the diameter of the ring would also be greater in size. So ito siya. So here you would have your media or your agar. Now your agar class would contain antibodies. In this case, antisera siya, which would have a target against your antigen of interest. Now, you would create your well or your sampling well. Yung well na yan, bubutasin nyo siya class, you would basta make sure na circular siya. Then that is where you would add the sample that would contain the antigen of interest. Now, you would wait upon adding the antigen class, a precipitation ring where the antigen in the sample would react with their specific antibodies in the agar, forming a so-called ring precipitation. So yan yung single radial immunodiffusion niya. So this is a more direct explanation of what happened, class. So yung antibodies that are found in the gel would spread throughout the antigens. Pupunta siya dun sa antigens ng well na ginawa nyo, forming a precipitate. So kung ito yung well nyo, upon adding the antigen, magkakaroon ka ngayon ng circular or ring precipitin line. So the positive result here would be a ring precipitin. Yan. Now there are two methods for single radial immunodiffusion, class. We have your endpoint method known as the Mancini technique, and we have your kinetic method known as the Fahey-McKelvey technique. So to differentiate them, class, yung Mancini technique nyo, please take note, would use measurements taken after the endpoint of equivalence is reached. So yung Mancini, you would only measure the ring precipitin after the endpoint of equivalence is reached. This would occur, class, between 24 to 48 hours if you're going to use an IgG antibody, and 72 hours if you're going to use an IgM antibody. The square of the diameter is directly proportional to the concentration of antigen. So the bigger yung class, yung ring na na-form, the larger or the greater the concentration of the antigen. Then for Fahey-McKelvey technique, class, measurements are taken before reaching the endpoint of equivalence. So readings are taken at about 18 hours. The diameter of the ring is proportional to the log of the antigen concentration. Then we have your double radial immunodiffusion technique. Now this is also known as your Auchterlonie double angular immunodiffusion. In this method, class, both antigens and antibodies would diffuse independently through agar gel in two dimensions, horizontally and vertically. Take note of that, class. In this method, yung pag-diffuse ng antigen and antibody nyo would be in two directions, both horizontally and vertically. As they diffuse radially, their concentration would diminish. At the point in the diffusion where the relative concentration of antibody and antigen would reach an equivalence, a precipitin line would appear. The technique is named for Orjan Auchterlonie, the Swedish physician who invented the test in 1948. Ito yung procedure niya, class. So you're going to perform this by cutting wells in the agar gel poured on a glass slide or in a Petri dish. The antibodies are placed in a center well. Take note, antibodies are placed in a center well and different antigens are added to the well surrounding the center well. After an incubation period of 12 to 48 hours in a moisture chamber, the precipitin lines are formed at the site of the combination of antigen and antibody. So, nasaan ba yung picture niya? Okay. Walang picture. Okay. Wait lang. Wait lang, class. Ito siya. So this is your Auchterlonie technique, class. So you're going to pour your agar in a plate. Pure concentrated agar, class. Walang antigen, walang antibody. Now, once you pour the media or the agar into the plate, you're going to create or you're going to make wells. On the center well, class, yung gitna na yan, that is where you would add your antibody or your antisera. Then you would make you would make wells surrounding the center well. Surrounding the center well class. And that is where you would add your antigens. Now, upon adding your antigens and your antibody, there would be three possible results that would form, known as your identity, non-identity, and partial identity, which I will explain in the PowerPoint. Okay. So as I mentioned, class, there are three possible results that would form upon adding your antigen and antibody into the different wells form. The first one would be your identity. And the second one would be partial identity. And the third one would be non-identity. So let's discuss them one by one. Let's start with your identity class. Now, identity class would form a fusion of lines at their junction to form an arc. So in this case, class, ito yung center well nyo. This is where your antibodies are located. Now, here you would add your antigens. What happens here, class, is that your antigens would move toward the antibody, and similar, class, your antibodies would move towards the antigen, forming an arc. Now, in this interpretation, the serologic identity or presence of common epitope, meaning they have two identical antigens. To simplify, class, let's say ang test nyo is you want to identify if the patient is positive for salmonella. So you would use the patient's serum. Now you would use here Salmonella typhi and Salmonella paratyphi antigens. Now, both of them, class, are somewhat similar in their epitope. Familiar kayo sa epitope, di ba? Epitope, class, di ba, ito yung antigen nyo. On the surface of the antigens, you would find these receptors. These receptors, class, are known as your epitopes that would wherein the antibody would attach. Now, since both Salmonella typhi and Salmonella paratyphi have the same receptors or have the same epitope, they would form an arc. That's why you would interpret this class as having identical or identity. Kaya siya tinawag na identity is because they have identical epitopes or antigen receptors on their surface, forming a junction or an arc. Ayan siya. Kapag naman partial identity, class, this is a fusion of lines with a spur. Pag sinabi mong spur, sumobra yung line, class. Sumobra yung line, yung radial line niya. Ang interpretation naman, class, is that the partial identity or there is a cross reaction. The spore form always points to a simpler antigen. So in this case, let's say again, ang patient nyo is positive for salmonella. Ang sample mo for antigen would be Salmonella typhi. Yung isa naman, Shigella dysenteriae. Of course, class, ang magpo-form ng junction dyan or yung magre-react na antibody dyan would be your Salmonella typhi. And that would be the one that would form a junction. While your Salmonella dysentery won't be able to form an arc since hindi naman siya specified or hindi naman positive yung patient for that type of disease. Kapag naman non-identity class, this is the presence of cross-line. This would demonstrate two separate reactions or the compared antigens would share no common epitope. In this case, class, non-identical yung antigens nyo. Magkaiba talaga sila. Let's say positive yung isa is S. aureus. Yung isa naman S. pyogenes. Now, yung patient nyo, class, is both positive. Both positive for this. Meron siyang what they call this? Meron siyang strep throat. Meron pa siyang skin disease or meron siyang boil. So pag may boil ka, there's a possibility na positive ka for antibodies for aureus. And kapag may strep throat ka, it's possible that you would also be positive for strep pyogenes. Now what happens here, class, is that your antibodies would go directly to each reaction, forming a cross line, because they would not share or there would be no common epitope. Hindi sila makakapag-meet in between. Now let's go to the precipitation by electrophoretic techniques. So this time, class, you're going to use electrophoresis. So this is a method that would separate molecules according to difference in their electric charge when they are placed in an electric field. A direct current is forced through the gel, causing antigen, antibody, or both to migrate. Now here, class, you would be using immunoelectrophoresis, which is a process that would combine, immunodiffusion and, mapabigay ako, that would combine immunodiffusion and electrophoresis. So examples of immunoelectrophoresis, class, is one-dimensional immunoelectrophoresis, two-dimensional immunoelectrophoresis, immunofixation electrophoresis. Now, under one-dimensional electroimmunodiffusion, we have your rocket immunoelectrophoresis. Then we also have your countercurrent immunoelectrophoresis. For the two-dimensional electroimmunodiffusion, there would be your cross immunoelectrophoresis. Now let's start with the one-dimensional rocket immunoelectrophoresis. So this is a modification, class, of your single radial immunodiffusion. This was developed by Laurel. It is called so due to the appearance of the precipitin bands in a shape of a cone-like structure, a rocket appearance at the end of the reaction. The height of the rocket precipitin is directly proportional to the amount of antigen in the sample. So the greater the height of the rocket precipitin, class, the more antigen present in the sample. Mainly used for the quantitative estimation of antigen in the serum. Now the principle here is that antibody is incorporated in the gel and the antigen is placed in wells cut in the gel. Electric current is then passed through the gel. A negatively charged antigen is electrophoresed in a gel containing antibody, which facilitates the migration of the antigen into the agar. This would result in the formation of the precipitin line, which has the shape of a rocket. The height of the rocket measured from the well to the apex is directly proportional to the amount of antigen in the sample. So ito siya, class. So this is your rocket immunoelectrophoresis. So for every well, so ito yung electrophoresis nyo, you would add your gel slab. Sa gel slab, class, maglalagay kayo ng, or may mga wells dyan. In the wells, class, you would add your antigen. And then in the gel would contain antibodies. Now, yung negatively charged antigens would migrate towards the anode. Yung anode, yung class, this is the positive electrode. Huwag kayong malilito dyan, class, kapag anion, negative ion. Kapag cation, positive ion. Kapag naman sa electrode, anode and cathode lang yan. Kapag cathode, negative electrode. Kapag anode, positive electrode. So if you have a negatively charged or an anion, anions will move to the anode or the positive electrode. In this case, yung antigen nyo, which is a negatively charged particle, would move towards the anode or the positive electrode. So ito, class. So maglalagay ka ng antigen dyan sa bawat well. Then you would run your electrophoresis. Yung electricity would be able to cause the diffusion of the antigen towards the antibody. So the greater the higher the rocket shape precipitation zone, the precipitation zone, the more antigen present. So ulitin ko ha, the higher the height of the rocket precipitin line, or the greater the antigen present. So that's your one-dimensional rocket immunoelectrophoresis. So if you, this is an example of a live, ang ganda ng pagkakaano nito ko, so pagkaka-diffuse niya. Sana magawa natin to sa lab kaso walang gamit. Okay, let's go to the next one, yung one-dimensional countercurrent immunoelectrophoresis. So this is an Auster-Loney technique that is enhanced and made quicker by using an electric current to bring the antigen and antibody together. Both antigen and antibody are placed in wells in an agar gel and made to migrate towards each other by electrophoresis. Diba? If you remember sa Auster-Loney, gagawa ka ng plate, you would pour your media, then at the center well, andun yung antibody, and surrounding the wells would be you would also create wells and you would add antigens. Dito class, same lang din yung principle, but this time you would use an electrophoresis. pH of buffer is set at 8.6 class so that the antibody is positively charged and the antigen is negatively charged. So by setting the pH at 8.6 class, yung antibodies nyo would become cations while the antigens would become anions. So ito yung principle niya. Antigen is placed in the cathodic well or the negative end, and antiserum is placed in the anodic well or the positive end. Antigen would migrate towards the anode while antibody would migrate towards the cathode. And precipitation would occur at equivalence within 30 to 60 minutes. So ito siya. You would add, di ba, ang antibody nyo. Your antibody nyo is a positive or a cation. Si antigen nyo would be an ion or a negative ion. So you would place class your antibody sa anode position, sa other side, sa positive electrode side. And then you would add your antigen, which is an ion, to the cathode. Now, what happens here is that by running the electrophoresis, both the antigen and the antibody would get attracted towards the middle part of the gel. Sa gitna sila magmi-meet, forming a precipitation line at the middle. So that's your one-dimensional countercurrent immunoelectrophoresis. Now let's go to the two-dimensional cross immunoelectrophoresis. So for the two-dimensional immunoelectrophoresis, this is also known as your cross immunoelectrophoresis. This is a variant of a rocket electrophoresis class. So this is a two-stage or two-dimensional procedure. The first dimension class or the first procedure is that antigens in solution are separated by electrophoresis. And for the second dimension, electrophoresis is carried out again, but perpendicular to that first stage to obtain rocket-like precipitation. So here you would have your antigen. Ito yung first dimension electrophoresis nyo class. You would first separate your antigens based on their size using electrophoresis. After that, you now add your antibody containing gel. Then you perform your second dimension electrophoresis and you would look for precipitin rockets. Diba pag ang antigen nyo, na-separate nyo siya, your antigens or your proteins would typically be separated according to size. Kapag mas pabilis or mas smaller sila class, they would tend to be found farther from the starting point. So after that, you now add your antibody containing gel. Pag na-separate nyo na yung antigen sa size, you now add another layer of gel. Then you run again another run, you would perform another electrophoresis, or the second step. Then you would now look for precipitin brackets. So ganyan ang magiging result ng gel nyo after running the second electrophoresis. Then we have the third one, yung immunoelectrophoresis nyo. So this is a double diffusion technique that would incorporate electrophoresis to enhance result. So ano lang din siya class, yung identity pattern, non-identity and partial identity. Now, if you take a look this time, instead of allowing it to stand for hours, mas mapapabilis yung result nyo by using an electrophoresis. So the source of antigen would be your serum, which is electrophoresed to separate out the main protein. Diba sa serum nyo class, if you still remember sa CC1 nyo, you would separate, if you perform electrophoresis on serum, they would be separated into different fractions. Yung albumin, yung alpha 1, alpha 2, and your gamma globulins or yung mga antibodies nyo. So you would perform electrophoresis on serum to separate the main protein. So tanggalin mo yung gamma globulins, tanggalin mo yung alpha 1, alpha 2, and even your albumin. Now once na-separate na yan, a trough is then cut in the gel parallel to the lines of separation. Antiserum is placed in the trough and the gel is incubated for 18 to 24 hours. Double diffusion would occur at a right angle to the electrophoretic separation, and precipitin lines develop where specific antigen-antibody combination would take place. Interpretation is difficult class, therefore it has largely been replaced by immunofixation electrophoresis, which would give faster result and is easier to interpret. So ito yung immunoelectrophoresis nyo class. So an antigen preparation, yung orange nyo, is first electrophoresis. So yung serum nyo, which is the source of your antigen, would undergo electrophoresis. Ang purpose nun class, as mentioned kanina sa previous slide, is that it would separate the antigens from the other major protein components. So kapag na-separate na yung mga antigens nyo based on their size, you would now cut a trough, yung parang trench class. Gagawa ka ng parang butas na horizontal. There you would add your antiserum, itong kulay blue na to. Dito sa ito yung trough, ito yung trench. That is where you would add your antiserum. Now, one or both sides of the separate antigen, and you would allow them to diffuse. So you incubate them for 18 to 24 hours class. Then you would wait for the antibodies to diffuse or mag-move towards the antigen. In time, lines of precipitation, itong mga to class, itong mga arcs na to, would form where the antibody and antigen would interact. So yan siya. So this is the step para mas maintindihan nyo pa. Trough would be cut in an agar. There would be the slide containing plain agar. Antigen would be placed in a well. Then you apply electric current. So pag in-apply mong electric current, masiseparate yung antigen source nyo dun sa serum. They would be separated in different components. Then you fill the trough with antibody. Antibody would diffuse towards the antigen component. Precipitin bands would form where antibody and antigen meet at optimal concentrations. Then we have another one, yung immunofixation electrophoresis niya. So in this case, let's say you perform serum protein electrophoresis and this is the pattern you would obtain. The pattern indicates abnormality in the gamma region of the sample. Now the question here, is it monoclonal or polyclonal? So kapag may abnormality ka class sa gamma region ng serum mo after separation, this is a case of monoclonal, but you need to know the specific answer, or you need to know what type of immunoglobulin or antibody is the one that has an abnormality. Is it your IgG? Is it your IgM, IgA, IgD, or IgE? In that case, class, you need to perform immunofixation. So ulitin ko class, let's say nag-decide ka mag-electrophorese ng serum ng patient mo. After performing electrophoresis, nagkaroon ka ng problem sa gamma globulin pattern. Now, you want to identify if this is a monoclonal or polyclonal problem. Now, this is a monoclonal problem class, but you want to identify which antibody ba yung may problema. So, is it IgA, G, A, M, E, D? So, you need to perform immunofixation. Now, after obtaining the serum protein electrophoresis pattern, protein fixative and monospecific antisera to IgG, A, M, kappa, and lambda are applied onto gel and incubated after. Now, you may be wondering, sir, bakit IgG, A, M, and G lang and kappa and lambda lang? What about your IgE and IgD? Remember, class, ang IgE nyo and IgD are rarely found in the serum. They are not considered present most of the time, class, sa serum nyo. So this is the procedure, class. So you would separate first the serum proteins based on their pattern, and then after that, class, you would add monospecific antiseras. So mag-add ka ng anti-IgG, anti-IgA, M, kappa, and lambda. Then you would run electrophoresis. Now, during incubation, insoluble immune complexes are formed. Then after incubation, the gel is now washed, pressed, dried, and stained. Patterns are then examined. So these are the possible results, class. Now, kapag yung specific antibody, nagkaroon siya ng pattern, katulad nito. These two are most likely the cause or the problem. In this case, it's either your IgG or the kappa region of the antibody is the problem. Monoclonal, this is, if you interpret this, class, this is a monoclonal gammopathy with increased IgG kappa protein. So this is your review lang, class, on a polyclonal versus monoclonal pattern. Now let's go to complement fixation. So for complement fixation, class, this is based on the fact that when antigen combines with an antibody in the presence of your complement, the complement is fixed by the antigen-antibody complex and is unable to react with cells synthesized with other antigen-antibody complex. Now, in your lecture class, naturo di ba yung complement? Ang complement nyo, these are a group of proteins. These are a group of proteins that would attach to the Fc portion of either IgG or IgM. Di ba ito yung antibody nyo? Itong dalawang to, these are the Fab. Itong part na to, this is your Fc. So dito mag-a-attach yung complement nyo. Now in complement fixation, class, you try to fix. When you say fix, hindi siya pwedeng mag-react with other antigen-antibody complexes. RBC sensitized with specific antibodies are used as an indicator of the presence of unfixed complement. Lysis of these RBCs would indicate the presence of unfixed complement. Lack of hemolysis class would indicate that the complement is fixed by the antigen antibody reaction. So ang positive, please take note class, sa complement fixation, ang positive result is that there would be no hemolysis. You may be wondering, sir, bakit no hemolysis? Remember, ang complement nyo, for you to be able to identify if the complement is successful, is that there would be the lysis of the cell done by the so-called MAC. Ano yung MAC? MAC refers to the membrane attack complex. Naalala niyo yung C5b6789. Yan yung MAC nyo. Pag yung MAC na activate, successful yung complement activation, there would be lysis. And dito sa complement fixation test, you try to or you fix the complement. You stop the complement from activating. Therefore, your positive result would be no hemolysis. So this is the first stage class. So you have here your antigen plus your complement. You would use, let's say, your serum that would contain with antibody against an antigen. Meaning class, yung patient is positive. Now, you would add your antigen plus your complement samples. Now, since positive siya, positive yung patient dun sa antigen, you would find that the serum would contain antibodies. Now, what happens, since meron siyang antibodies na present class, there would be complement fixation. But if you add a serum without antibody, meaning negative yung patient nyo, negative yung patient, if you add the antigen and the complement, automatically maglalays yung antigen class. Masisira yung antigen or sisirain ng complement or the complement will destroy the antigen. Ito, para mas maintindihan nyo pa class. So in this type of case, ang reagent nyo that you're going to use as an antigen is your SHIP RBC. Now you add your antibody to the SHIP RBC. If there is no hemolysis, the reason for no hemolysis is that your complement is fixed to the antigen antibody reaction. So hindi makapag-activate yung complement nyo because it's already fixed towards the antigen antibody. Now if another example, ito naman negative result naman siya. Here you would have your antigen, SHIP RBC, then you add your antibody to SHIP RBC. Hemolysis would occur class because of uncombined complement available. So take note of the interpretation. Positive is that all available complement is fixed by the antigen antibody reaction. No hemolysis would occur, therefore the test is positive for the presence of antibody. Pag negative test naman, no antigen antibody reaction, the complement remains and the red blood cell are lies in the indicator stage so that the test is negative. Ayun class. So that ends the activity 3, yung precipitation nyo. So medyo madami din siya, somewhat similar with activity 2, pero mas siya class, mas madaming, mas complex yung mga tests when it comes to precipitation. And very difficult siya gawin in the laboratory. That's why sa mga tests nyo, when it comes to immunosero, immunohematology, most of the time ang principle is usually agglutination kasi mas madali yung reaction niya. Now, I'm going to explain to you what you would be doing for your activity 3 kasi hindi nga natin siya magagawa on-site.
Okay. Dito sa draw and interpret ng result class, you're going to draw the 3 possible results in an Auster-Loney double immunodiffusion assay. So ulitin ko ha, dito sa draw and interpret your result, you're going to submit or you're going to draw the 3 possible results in the Auster-Loney method. Okay? Ang deadline niyan would be on the exam day. So, i-collect ko lahat ng manual nyo. Make sure to answer the post-lab questions as well. So, again, for the last time, for activity 3, i-draw nyo yung 3 possible results ng Auster-Loney double diffusion. Then, don't forget to answer the post-lab questions. The deadline would be on the exam day. Ang exam natin, class, it would be on February 10. February 10 na lang tayo mag-exam. Okay, may question ba, class? Clarifications? Wala. Clear naman, no? Sige, next meeting, that would be on February 3. We'll meet on site. We'll have your quiz on activity 2. Then we'll do the activity 2 experiment. So kindly bring the necessary material